Hello friends, welcome to my channel. Here we'll study the 12th standard biology. Part 3 Reproduction in lower and higher plants. In this video we'll study the following points, that is pollination, types of pollination, agents, types of agents, and outbreeding devices. Pollination is a vital process in the sexual reproduction of flowering plants. It is the transfer of pollen grains from the anther, the male part of a flower, to the stigma, the female part. This process is called as the pollination process. The ways of pollination. The first one is self pollination. This happens when pollens from a flower is transferred to the stigma of the same flower or to another flower on the same plant. While this method is reliable and doesn't require an external agent, it can lead to a reduction in genetic diversity. The next one is autogamy, the transfer of pollen to the stigma of the same flower. The third one is Gaetonogamy, the transfer of pollen from the flower to the another flower on the same plant. The next one is the cross pollination or xenogamy. It means this is the transfer of pollen from different plant of the same species. Cross pollination propagates the resistance to diseases and environmental changes. Biotic agents, these are living organisms that carry the pollens. Example, insects called entomophili. These are bees, butterflies, and moths are the common insect pollinators, often attached by bright colors, fragrances, and nectars. Now, the next one is a bird, Omitophily. Birds like hummingbird pollinate flowers, often those with tubular shape and bright colors. The next is bats, Cremo, Chiropteromorphy. Bats are important pollinators, especially in tropical regions. Now we'll see the abiotic agents. These are non living factors that aid in pollination. Example, wind, anemophily. Wind pollinated flowers are typically small, icon-spacious, and produce large quantities of lightweight, non sticky pollens. The next one is water, hydrophily. This is less common but occurs in the same aquatic plants where pollen is carried by water contents. Pollen grains are microscopic structure that represents a male gametes of seed plants. They are essential for plant reproduction as they contain the male gametes. In classes, the study of pollen grains is a key part of the sexual reproduction in flowering plant. So here we'll see the structure of a pollen grain. A typical pollen grain is generally spherical and has a two-layered wall. The first one is exine. This is a hard outer layer made up of highly resistant substance called sporopollenin. The sporopollenin is one of the most durable organic materials known and is resistant to high temperature, the strong acid and alkali. This layer protects the pollen grain during its journey from the anther to the stigma. The exine often has interact patterns and design that are unique to each plant's species and punctuated by one or more prominent apparatus called germ pore where sporulin is absent. The next one is the intine. This is a thin inner wall made up of cellulose and pectin. It is a continuous layer that surrounds the cytoplasm. Inside the pollen grain, there is a cytoplasm surrounded by a plasma membrane. A mature pollen grain typically contains two cells. The first one is the vegetative cell. This is the layer of two cells. It has abundant food reserves and a large, regularly irregular shaped nucleus. Its primary function is to the pollen tube during germination. The next one is generative cells or generative cells. This is a smaller spindle shaped cell with dense cytoplasm and nucleus. It floats in the cytoplasm of a vegetative cell. The generative cell divides mitotically to produce the two male gametes. Development of pollen grains. The formation of pollen grains is a process known as microsporogenesis occurs within the anther of a flower. Here is a simplified overview of the process. Microspore or a mother cell. These are diploid cells found in the pollen sac of the anther. The next one is the meiosis. Each pollen mother cell undergoes mitotic division to produce a cluster of four haploid cells called microspore traits. The next one is maturation. As the anther matures and dehydrates, the microspore dissociate from the tetrad and develop into individual pollen grain. The next one is the mitotic division. The nucleus of the microspore divides mitotically to form the vegetative and generative cells. In some species, the generative cells divide again to form two male gametes before the pollen grain is shed. Three-celled stage. While in others, this division occurs later after the pollen grains had landed on the stigma. Significance of pollen grains. The male gamete carrier. Pollen grains are the carriers of the male gametes, which are essential for fertilization and the formation of seeds and fruits. The next one is the genetic diversity. Pollen transfer from one plant to another across the pollination. It helps to maintain and increase genetic diversity within a plant pollination. Now, the next one is the pollen allergy. Pollen from certain plants like Parthenium carrot grass can cause severe allergy and chronic respiratory disorders such as asthma and bronchitis in some people. Pollen banks, due to their important and plant breeding programs, pollens can be stored in pollen banks for longer period in liquid nitrogen under minus 190 minus 196 degrees Celsius. Self pollination. It is a form of pollination where pollen is transferred from the anther to the stigma of the same flower or to another flower on the same plant. This process is not required a pollinating agent from the different plant, making it reliable method of reproduction. It is a key topic, okay, of reproduction in plants. Now we'll see the types of self pollination. Self pollination is categorized into two types. The first one is autogamy. In this process, this is a transfer of pollens from the another to the stigma of the same flower. For autogamy to be successful, the anther and stigma must mature at the same time, a condition known as homogamy. Some plants like wheat and pea have flowers that remain closed, a condition called clistogamy, ensuring that only self pollination occurs. The next one is gatonogamy. This involves the transfer of pollination from the anther of one flower to the stigma of different flower on the same plant. Although this process often involves external agents like winds or insects, it is genetically considered self-pollination because the pollen and the ovule comes from the same plant, meaning the offspring will be genetically identical to the parent. An example of this is maize, where pollen from the male tassel pollinates the female silks on the same plant. Xenogamy. It is also known as the cross pollination in a true genetic sense. It is the transfer of pollen grains from the anther of a flower on one plant to the stigma of a flower on a different plant of the same species. This is a crucial process of the pollination process that results in a new genetic combination, promoting a genetic diversity within a species. Now, the key features of the xenogamy are as follows: the genetic variation. The most significant outcome of xenogamy is the production of genetically distinct offsprings, since the pollen of the ovule comes from different parent plants. The resulting seeds combines genetic material from two individuals. The genetic diversity is vital for long term survival and evolution of a species, as it increases the population ability to adapt the changing environment, diseases and pests. Now we'll see the outbreeding devices. Many plants has evolved species mechanism called outbreeding devices to prevent self-pollination and encourages xenogamy. These mechanisms include dichogamy, hicogamy, self incompatibility, and diocy. Now we'll see the first. The dichogamy means the anther and stigma of a flower matures at a different times. The next one is hercogamy. The physical arrangement of the anther and stigma creates a barrier that prevents self pollination. The next one is self incompatibility. The pollen from a flower is unable to germinate on the stigma of the same flower or another flowering plant. The next is plant has the unisexual flowers with male and female flowers on separate plants. Example, papaya. This obligates the papaya plant to undergo xenogamy. The agents of xenogamy. Xenogamy almost always requires an external pollinating agent to transfer the pollen. These agents can be either biotic living organisms or abiotic non living factors. Now, biotic agents, insects like bees and butterflies, birds and bats are common biotic agents. Flowers that rely on these agents are often large, brightly colored, fragrant, and produce nectar to attract them. Now, the abiotic agents, wind and water, are the primarily abiotic agents. Wind pollinated flowers are typically small, inco- inconspicuous, and produces a large number of lightweight pollen grains. Biotic agents are per- of pollination are non-living factors that facilitates the transfer of pollen grains from the anther to the stigma. They are the primary method of pollination for a smaller portion of a plant compared to biotic agents, living organisms like insects or birds. The two main abiotic agents are wind and of water. Wind is also called as amniophily, and the water is also called as the hydrophily. The examples of wind are many grasses like wheat, rice, and maize, and the examples of waters are plants like tape grass, hydrilla, and some sea grasses. Pollination by water, also known as hydrophily. It is a rare and specialized form of abiotic pollination that occurs in some aquatic plants. This method is far less common than pollination by wind or animals, with only about 30 generation of aquatic plants using it. There are two main types of water pollination. First one is epinhydrophily, and the second one is hypohydrophily. Adaptations of water pollinated plants. Pollen grains. The first one is the pollen grains. The pollen grains are often lightweight and have a nucleus covering that prevents them from getting wet. In some cases, like Zostera, they are long and noded like to increase the surface area for contact with the stigma. Flowers. The flowers are typically small and lack of nectar and without fragrance, as their traits are not needed to attract the pollinators. Now, the wastage. Due to the random nature of water current, the vast amount of pollen is produced and released with very small percentage successfully reaching to the stigma. Hydrophily. This is the term of pollination that is facilitated by water. It is a relatively uncommon method, primarily found in some aquatic plants. The two main types of hydrophily are as follows. and these are categorized by the location where the pollination process occurs. The first one is epihydrophily. This is a type of pollination occurs in the surface of the water. Mechanism of this process are as follows. In epihydrophily, the male flowers or pollen grains are released and float on the water surface. They are then passively carried by water currents until they come into contact with the female flowers, which have also reached the surface. Plants adaptations. Plants that undergoes epihydrophily often have a mechanism to bring their flowers to the water surface at the right time. For example, the female flowers of eelgrass have a long coiled stalk that uncoils to bring the flower to the surface of the water. Example, a classic example is a eelgrass. The male flowers detaches and floats on the water surface, and some of them eventually reach and pollinate the female flowers. Hypohydrophily. This is a type of pollination occurs entirely beneath the surface of the water. Mechanism. In hypohydrophily, both the male and female flowers remain submerged. The pollen grains are released underwater and are either heavier than water, causing them to sink, or they have neutral neutral in nature, allowing them to drift through the water column. They are then caught by the stigma of a submerged female flower. Now, pollen adaptations. The pollen grains of hypohydrophil plant often have specialized features. They may be long and ribbon like, as in some sea grasses, to increase their chances of making contact with the stigma, and they are typically protected by a mucilaginous coating to prevent them from getting waterlogging. Examples of plant that undergo hydrophily includes Zoostera, seagrass, and Najas. Biotic agents are living organisms that facilitates the transfer of pollen grains from the anther of one flower to the stigma of another. This is the most common method of pollination in flowering plants. These agents are attracted to flowers by rewards such as nectar, pollens, fragrance, or a place to lay eggs. There are main types of biotic agents. The first one is insects called entomophily. This is the most prevalent type of biotic pollinations. Bees are the most dormant dominant insect pollinators, but butterflies, moths, flies, and bees are also play a significant role. Flowers pollinated by insect are often large, brightly colors and fragrant. They may also have specific patterns or making that guide the insect to the nectar. The pollen grains are often sticky or spiky, making them easy to attract to the insect body. The next one is bird, also called as ornithophily. Now we'll see here this occurs in a variety of plants, especially those with a large sturdy flowers. Birds such as hummingbirds and sunbirds have a high metabolic rate and require a lot of energy. So they are attracted to flowers that produces a little amount of nectar. These flowers are typically tubular in shape, brightly, often red, orange, and yellow. The lack of a strong scent, as birds have a poor sense of smell. Now, the third one is bat. Chiro, Chiropterophily. This type of pollination is common in tropical and subtropical regions. Flowers pollinated by bats typically bloom at night, as bats and are nocturnal. They are often large, light colored, or white and have a strong fruity or musky odor to attract bats. The flowers also have produced a large amount of nectar to sustain the bats. Other animals called zoophily. While less common, other animals like some primates, example lemurs, rodents, and even lizards have been observed pollinating flowers. These animals are typically attracted to large flowers within high volume of nectar. Outbreeding devices are a set of mechanisms developed by flowering plants to prevent self-pollination and promotes cross-pollination. This is an important strategy because continuous self-pollination or interbreeding can lead to a phenomenon called in in breeding depression, which results in a reduction in vigor, fertility, and overall health of the offspring due to the increased expression of receivers deletion genes. The main type of outbreeding devices are dichogamy. This is the most common outbreeding devices and involves the male and female reproductive organ of a flower maturing at different times. This prevents the pollen from the same flower from fertilizing its own stigma. Dichogamy can be of two types. The first one is protandry. The anthers or male parts mature and releases pollen before the stigma or the female part becomes receptive. Examples including sunflowers and maize. The next one is protogeny. The stigma matures and becomes receptive before the anthers releases pollen. Examples including some species of fig and millet. Now here we'll see the next second one, that is unisexuality, called declining. In this case the flowers are unisexual, containing either only male staminate or only female pestillinate reproductive organs. This completely prevents the autogamy or self pollination within the same flowers. The monoecious plants. We'll see here the example monoecious plants. Both the male and female flowers are present on the same plant, while autogamy is prevented. Gaetogamy, called pollination between different flowers on the same plant, is still possible. Now here we see the examples includes the maize and the castor. Now next one is dioecious plants. Now we'll see here what is mean by the dioecious plants. Male and female plants flowers are on separate plants. This prevents both autogamy and gaetogamy, ensuring that only